Meiosis I, people. If you know what you're doing, this is review for you. Fast forward to like 45 seconds. But if not, drawing some cells. We've got the dotted line represents the division between the cell before meiosis begins and the 4 cells we see during meiosis I. I'm just going to label these up here a little before meiosis here. And then prophase 1, metaphase 1, anaphase 1, and telophase 1. I know, I screwed that up. I'll erase it. All right, let's zoom in. Let's take a look at the nucleus. There's your nuclear membrane. Look at that DNA, nice and thin. Nice and thin, that's chromatin, people. I'm going to use two colors, because this individual got half its DNA from mama, half from the papa. Now, in prophase, scratch that. Let's get those details in there. See those centrioles sitting, hanging out right near the nucleus. Label up the chromatin. Now we're on to prophase. Notice that nuclear membrane, she's breaking up. Let's draw up some of that DNA nice and thick. First thing is that DNA is coiling into chromosomes. Why is it doing that? Hey, Jimmy, it's doing that because it's got to move. You can't move chromatin. Chromatin too thin, messy. So we got homologous chromosome pairs grabbing together. And we still have some DNA hasn't quite coiled, but by the end of prophase, all that DNA is going to be in the chromosome form. All homologous chromosome pairs will be joined together. And notice the crossing over between the homologous chromosomes, the non-identical sister chromatids of the homologous chromosome pairs. Every single one of them has the opportunity to cross over at least once. Mucho shuffling of the deck there. Now on to metaphase. Our homologous chromosome pairs are going to line up across the middle. See that they now have swapped pieces from the crossing over. The blue chromosome that came from Papa, it's got a little bit of Mama's chromosome on there. Same thing from the Mama. Now I'm going to go for broke here. I'm going to have these little guys cross over as well. Mr. Gnomes, I thought humans had 46 chromosomes. Why are there only 8 there? Hey, Jimmy, this is just a fictitious organism. I am not drawing 23 pairs of chromosomes in this. How much time do you think I had to kill? Now look at the spindle fibers. Some of them go right from centriole to centromere, then some of them overlap. Key, both of them, key players in what happens in anaphase. Look at that long cell. It got elongated because of those overlapping spindle fibers. They overlap and tiny little motor molecules push against the opposite centriole, spindle fiber, making the centrioles and hence the cell get further away from each other. The ends of the cell get further away from each other and making that cell that egg shaped. This helps separate the chromosomes, helps have much more room for when the pinching begins in telophase. You're going to ensure or increase the chance that chromosomes have crossed over. Now we see what happens here is the homologous chromosome pairs, they have separated. We see that big red guy on the left has dropped down, and the little red guy on the right has dropped down. Give them their little bits of blue. And then the blue guys from each of those homologous chromosome pairs, they have gone up. The spindle fibers have brought them there. I use the word brought because it looks like they're being pulled. There's some pulling, there's some pushing. No need to get into that. On to telophase. Notice the most obvious peanut shape of the cell. That's because of a protein fiber that is growing around the membrane, cell membrane. Technically, it's weaving in and out of it. And then we see those nuclear membranes reforming. How they do so, yo no sé. Spindle fibers coming up. Notice how much shorter they are. Some growing from each of the centrioles, little bits and pieces, they're disappearing. We don't need them anymore. And then some of them still in the middle. We'll see that if we look at cells in the middle of mitosis or meiosis in a slide. And then our DNA, it's uncoiling. So we're going to see some DNA in its chromosome shape and some in its chromatin shape. So that's why I'm just doing some thick, some thin, because it's got to get itself back to chromatin. It needs to be in chromatin. Why? Because you're going to have to make another set of centrioles. Each of the cells that we're going to make after this pinches, after telophase, this will pinch during a cytokinesis event, and you'll end up with two cells. Each cell will only have one centriole. We're going to need a second centriole. We'll see in meiosis II, a totally different video, there'll be two centrioles per cell. You need that chromatin in that shape so it can be read. Remember, chromatin is easier to read. Chromosomes are easier to move. Here's my pièce de résistance, my 3D drawing of that protein fiber. You all know how proud I am of my artistic skills drawing that. Now let's add in some verbiage to make sure we emphasize homologous chromosomes roles, crossing over, and the homologous chromosome separating an anaphase. So that's meiosis I. On to meiosis II.